Welcome to second semester physiology. So this is a special course for physiology where in the first 6 lectures, inshallah, we will be revising some of the physiology that you have already done in the first semester. So that is why it will be relatively easy. Then inshallah I will start with the nervous system. So last time you were with me and I taught you kidney, renal system, etc. and that was very interesting. This time maybe some slides they'll be boring or something like this, but inshallah I will try my best to keep you interested. Now, first we start with the same question. Do you remember what is physiology? And in physiology, what do we learn? So physiology, it is, as you know, it is the knowledge where we discuss the functions of the body, how the body performs its function. So for example, I put an example which will make our task easy. It is the study of the normal functions of living organisms and their body parts. And it tries to answer 2 questions. Number 1, what is the purpose of a body process? And number 2, what is the mechanism of action? For example, if our heart rate increases, what is the purpose of heart rate increase? It is obviously to increase the blood flow to the tissues. Number 2, how the heart rate increases. What is the mechanism of action of increase of heart rate? So for example, you can tell me that the sympathetic stimulation or adrenaline, like when we are afraid of something, so it increases the heart rate. So we discuss the purpose of a body process and we discuss the mechanism of action of a body process. Is it fine or I put off the light? It's fine. Alright. So I've got a few examples here. For example, process. Why do I shiver? Because this is now we have cold. So shiver. Shiver means,
ارتعاش.
Yes. Saleh, what will you say this in Arabic?
ارتعاش.
So why do I shiver when I am cold? When a person feels very much cold at Fajr time, for example, he's going to masjid and he feels cold and his body starts shivering. So this is to help warm up, because shivering, it breaks down ATP, the muscles they contract and we generate heat. Then why there is shivering? How there is shivering? What is the mechanism of shivering? So what happens is this, that when the temperature falls, it is recorded by the receptors, which are called thermoreceptors, which are there for recording the temperature. Then the nerve signals, they go to the brain, the area which controls the temperature, and I will be discussing it with you inshallah in another 10 minutes. And the nerve signals from this center in the brain, they go to the muscles, and the muscles, they start irta'ash or oscillation or what we call shiver. So this is physiology. That is, we discuss a mechanism and the process. What is the purpose of the process? I go back to this slide. And what is the mechanism of action of any process? So I have a couple of examples. For example, a list of our topics in the second semester in first unit introduction and the detail of the syllabus of introduction. And we start with the first thing which you have learned in the first semester, that is homeostasis. And I will just pass through these slides in an organized fashion, inshallah. Now, if someone asks you what is homeostasis, so homeostasis, it is the maintenance of the internal environment of the body. Now, what is meant by the internal environment of the body? See, if we are in this room, so I am Ahmed, he is Muhammad, he is Mahmoud, he is Saleh, everyone is different. But our external environment, this is common, How much oxygen is there, how much carbon dioxide is there, what is the temperature here, anything, bad smell here, we all feel it. But what is inside Muhammad, this is his own. What is inside Mahmoud, what is inside Saleh, what is inside Ahmed, this is own. So just like that, our body cells, stomach cells, brain cells, pancreas cells, thyroid cells, kidney cells, they all live in an environment which is extracellular fluid, So what is meant by the internal environment? So internal environment is not what is inside the stomach, or what is inside the thyroid, or what is inside the muscle. It is the outside, or the fluid outside the cells, which is called extracellular fluid, which must be having maintenance of everything. If it is not maintained, we get disease. Now, the concept of external and internal environment of the body, as I told you, for example, this is the stomach cell. For example, this is the thyroid cell. For example, this is the kidney cell. But all of these cells, they are living in the internal environment, which is extracellular fluid. You see? Intracellular fluid, there is present. This is special. For example, stomach has HCl in it. It has got different pH, which is different from this place. Thyroid has got more iodine, which is different from this place. So this place is the common place. So we say internal and external environment. When we talk about homeostasis, we say that homeostasis, it is the maintenance of the internal environment of the body, which is basically the extracellular fluid. So please, everything as you have learned in the previous exam, for my 2 parts which I had with you, everything came from the slides. So inshallah, trust me, everything will come from the slides. What is external environment? It is defined there. What is internal environment? It is defined there. So we must have a clear concept of the external and internal environment of the body. So we have to maintain many things. For example, our temperature must be maintained. For example, our pH must be maintained. Fluid and electrolyte must be maintained. How much sodium is there, how much potassium is there, how much magnesium is there, it must be controlled. How much water is there, this must be controlled. You know, if we have less water, this is called dehydration. And we need water then. If there is more water, this is called overhydration. And we need to remove the water from the body. So maintaining the balance, this is called homeostasis. Blood glucose must be maintained, and I will discuss the example of blood glucose in a minute. Blood calcium must be maintained, and there are hundreds of examples of the things which must be maintained. This is called homeostasis of the internal environment. Now there are 2 mechanisms which you must be remembering from your first semester. One we call negative feedback and the other we call positive feedback. So in the negative feedback, the stimulus and the response, they are in the opposite direction. And I have got straight away example, which first I will tell you and then I will go to the slide. For example, if I eat food, in fact I'll give the example of a chocolate. I eat a chocolate. So my blood glucose level will increase. So when my blood glucose level will increase, this is the stimulus, increase. Now, something happens, which I will discuss in a minute. Number 1, blood glucose has increased. What is homeostasis? It should be decreased, brought back to the normal. Yani, stimulus and response, they are in the opposite direction. In between, what happens when blood glucose increases? Signal goes to the pancreas, and pancreas secretes a hormone which is called insulin. So insulin, this hormone then causes decrease in the blood glucose level. How? The blood glucose enters into the cells because insulin opens the gate for blood glucose. So what I am saying is this: stimulus, increase in the glucose, and response, decrease in the glucose. So because they are in 2 opposite directions, therefore, see, so stimulus, increase in the blood glucose level, and response, decrease in the blood glucose level. So because they are in the opposite direction, therefore we call this as negative feedback mechanism. That's okay. So stimulus, it is increased blood glucose. Response, it is decreased blood glucose. What was the mechanism? It was insulin. So because these are two in opposite directions, therefore we say that this is negative feedback mechanism. This will be exactly opposite to the positive feedback mechanism, where the stimulus and response, both of them, they will be in the same direction. So we see a diagram. This person, he is eating food. So stimulus is increasing the blood glucose level. High blood glucose level leads to pancreas, which secretes insulin. This increases the take-up of glucose by the cells and by the liver. This leads to return of the blood glucose back to the normal. So this is example of the negative feedback. This in detail I have written here for you so that you can prepare it for your quiz, and I am not stopping here. This is exactly what I told. This is now new, that three things they were involved. One is sensor, which picked up the blood glucose level. One is processor, and one is effector. So these are the three components: sensor and the control center, and finally the effector. Sensor detects the change. Control center makes the decision that what has to be done, and the effector, it responds. So sensor receives the stimulus. Effector, this produces the response. Control center, this makes the decision. So this is what we call negative feedback mechanism. So as I told you, that positive feedback is exactly opposite to the negative feedback. That is, in the positive feedback, the stimulus and the response, both are in the same direction. So because they are going in the same direction, so this is opposite to the negative feedback, and that is why it is called positive feedback. So, for example, we have got a beautiful example which you have done before as well. That is the example of the birth of the baby. Now, baby stays in the mother's uterus for 9 months. All of you know it. And in those 9 months, it keeps on growing and growing and growing. So when a time comes, when the baby is sufficiently grown up and the uterus cannot become bigger, what happens is this: by that time, the receptors for oxytocin are there all over the uterus. Now, the step number one, head of the fetus, it pushes against the cervix. This is closed, subhanAllah. It is totally closed. Nothing can go inside. Nothing can come inside, come outside. So this is vagina, and this is the uterus mouth. This is rahim, uterus. You see? So baby's head, it pushes here. When it pushes here, it stretches. When it stretches, the nerve impulse, it goes to the brain, hypothalamus. When it goes to the brain from this, it leads to the stimulation of posterior pituitary gland, which produces a hormone called oxytocin. This hormone, it acts on the uterus and it causes contraction of the uterus. Now, when the uterus it contracts, baby head is pushed more. When it is pushed more, it stretches more. When it stretches more oxytocin, more contraction, more stretch, more oxytocin, more stretch. So what is the direction of the stimulus? It is stretching, increasing. What is the response? More stretching. So yani both stimulus and response are in the same direction. This is called positive feedback, and we have got very few examples of this. So just like negative feedback, I have put these points which I said here with some examples. Number one, birth of the baby. Number two, action potential. Number three, the blood coagulation. So these are the few examples of the positive feedback mechanism. and this is what we revised from our first semester. Now we have got another example of homeostasis, which is based upon the temperature regulation. And this is a big topic, a new topic, which you will prepare good because you can have multiple questions from this. Now, temperature has two components. One is heat and the other is cold. If, like today, if it is cold, I am wearing this coat to cover myself. You see, it is making insulation around my body. But you see the cat which is sitting outside, the cat, Kitta. Now this cat, it does not wear the coat. But does it have a system to insulate its body? Yes. Its hair stand up when it is cold, you know. When it is cold, what happens to us? We do not want to lose our heat. So there is vasoconstriction. The blood vessels, they constrict and the heat cannot be lost to the atmosphere. I go to the opposite. When it is hot weather, now our blood vessels, they become dilated so that they bring more heat to our surface, and if there is cold outside, yani our body temperature is more than the outside temperature, then this heat will be going out into the air, convection, or to the surface which is cold, conduction, physics. And if the temperature outside is more, what will happen? We will get sweating, and the sweating will be evaporated, the sweat, tabkhir. So this will cause cooling, you know. So Allah subhanahu wa ta'ala has made a system where we have got a control system in our hypothalamus in the brain. This is called hypothalamic thermostat. It is fixed on 37 degrees centigrade. Subhanallah. So this, if temperature goes down, the signals go to the brain, you know, which then causes constriction of the blood vessels throughout the body. So then, this constriction, it causes conservation of the body heat. So body heat is conserved. Same time, this causes shivering. When there is shivering, there is heat generation. So both of these, they cause increase in the body temperature. Just like that, if the temperature is more, the signals go into the brain, which causes vasodilatation in the skin, and it causes the sweat glands to secrete, and the body heat is lost and the temperature goes down. So these are two methods, one to increase the temperature, the other to decrease the temperature. Both are called temperature regulation, and this is an example of homeostasis. So we will go into a little detail of this topic, that is temperature regulation. Now first, where is the center for temperature regulation? This center is in the hypothalamus. So Saleh, can you put your camera to this side? See, this is the thermostat. This is the thermostat. Yani, I can increase or decrease the temperature of this room. If I want this room to be at 30, I will put it at 30. If I make it down 15, this room will become very cold. So just like that, Allah subhanahu wa ta'ala has made a thermostat in our hypothalamus. You know who can disturb this? The bacteria can disturb it. The bacteria, when they cause infection in our body, they disturb our hypothalamus. They disturb this thermostat which is fixed at 37, and they cause us fever, which I will tell you in the next slide. So this is in the anterior hypothalamus. If the temperature goes above 37, this causes sweating. If the temperature goes below 37 up to 36, this is causing just vasoconstriction, so that heat is not lost from the body. If the temperature goes down to 35, this causes shivering. You see, so this is hypothalamus, which is controlling the temperature. And what is the set point of hypothalamus? The set point of hypothalamus is 37. This I told you in the previous semester as well. Please prepare the figures also. The question comes from the figures also. Do not leave the figures. So I have put it here. What is the normal body temperature? 37 degrees centigrade. What is this? This is called hypothalamic set point. So Saleh, do you know what is hypothalamic set point? How much is it?
37.
37 degrees centigrade. Alright. So now, based upon this, we give one good example and one bad example. The good example is this, that you are playing football or you are running and exercising. So your muscles, they are breaking ATP. When they break ATP, the heat is produced. This heat stimulates your hypothalamus. This causes sweating on your body. Sweat is then evaporated and your body temperature, it becomes normal. Can you believe when you are playing a football match, your temperature is not 37. It may reach up to 39. You know, because your muscles, they are active and they are breaking ATP. But subhanAllah, immediately this is brought down by our normal body. But there are many mechanisms, and we will have a comparison of them. Prepare this for your MCQ, prepare this for your extended matching question. That is, what are the responses of the body to the heat? And I told you all of them just now. Number 1, vasodilatation. Number 2, sweating. Number 3, these hair, they get relaxed so that there is no insulation. Number 4, we like to stretch out like this. We don't want to be like curling up like a cat, which we do in the cold. You know, in cold we sit together. In heat we say, Yani, you go a little further. It's hot. Stay away. And I like to sit like this. So in cold there is vasoconstriction. In heat there is vasodilatation. In cold there is shivering. In cold there is piloerection, yani the hair stand up. And in cold we like to be flexed. This is called curling up. This is called curling up. Now, what will happen if temperature regulation fails? Saleh?
Yes.
Can you tell what will happen if the temperature regulation fails?
Fever.
Yes, it will be fever. Yani our body temperature, it will increase. So it can be fever. What will happen if it goes to the opposite side? It will become hypothermia. Yani our body will become very cold. Like many people, they die when there is snow in Canada. If they are traveling on the road and the car brakes, what can they do? The temperature is minus 40. So they get hypothermia and they die of hypothermia. So we'll take example of the fever. So fever, when more heat is generated, then heat loss. Heat exhaustion, when we are exposed to heat. Sunstroke, darbush shams. Sunstroke, darbush shams. Exposure to heat. I am standing in Arafat, and I am standing in Arafat in summer, and there is sun, and I am standing outside and praying for a long time and not drinking water and not cooling myself. So I get darbush shams. So this is sunstroke. So they are the examples of increased temperature. What happens if they become very much cold? This is called frostbite. Frostbite. So this is due to the exposure to the cold. So what is the fever? How the fever develops? So as I told you just 2 slides back, that is hypothalamic thermostat, it is set at 37 degrees centigrade. When this is reset, yani someone makes it 38 or someone makes it 39, this means the body takes the body temperature to the new set point. Like if I go to that thermostat and I increase it to 30, now this means the AC cooling will stop. Maybe heating starts so that the temperature of the room increases. You understand? So when the bacteria, they make it from, for example, from 37, the bacteria, they make it 39 or they make it 40. So what will be the new temperature for the body? This will be the new normal temperature for the body. What will body do? It will cause increased ATP breakdown. So ATP breakdown will be more. So what will happen? More heat will be produced. What will happen? Our body temperature will reach, for example, 39. When it becomes 39, we will be happy. All of you got fever fever sometime in your life. When you get fever, first you feel chills. Then your mother brings a blanket and puts on you. Then your temperature increases. When it reaches the new normal which the bacteria has made, you become happy. But actually you are not happy. Your body is breaking down. Your enzymes are breaking down. It's like you put an egg into hot water. So the proteins in the egg, they become, boiled. They become white. You understand? So breakdown of the protein. So now, what is the mechanism of fever? This is, I am just naming bacteria, but there are multiple factors which the bacteria produce or something else it produces. So I'll give you a couple of names here, and they are easy to remember, and remember them for your life. Now these are the different mechanisms. Elevated set point of hypothalamic thermostat will cause fever. Entrance of pyrogens, for example, bacteria and viruses can cause fever. What is pyrogen? Pyrogen is a thing which can cause fever. Alright? Pyrogen is a thing which can cause fever. What is the second name for fever? Fever is equal to pyrexia. Pyrexia. Sahi? So, yani fever and pyrexia. So, from this we have got this new word, pyrogens. So, pyrogens are the substances which cause pyrexia. What is pyrexia? Pyrexia is fever. Okay? So, can you give some example of pyrogens? Yes, bacteria and viruses. And maybe fungus, maybe other things. Many things we do not know. Many things have not been discovered. So this is about the fear. Then what next? Disruption of hypothalamic thermostat, like this one. Increased heat production. For example, chills, skeletal muscle response. So this is leading to fever. Decreased loss of heat from the body. This can lead to fever. So these are the different mechanisms by which fever can take place. Now we change the topic, and we have covered our first topic, that is homeostasis and the example of the temperature regulation. Now we go to the transport across cell membrane. And you know why I have put this diagram over here? Saleh, any idea? You see, there is a difference. This person is coming down. And this person is going
up.
Which one will need energy? This or this? This one. This one will need a lot of energy because he is going uphill. Will he need more energy? No, because he is going downhill. What mechanism of transport comes into your mind if I show you this diagram? Diffusion. From high concentration to low concentration. No energy used. What mechanism comes into your mind by this? Active transport. Active transport. Yani it will need energy or ATP to go up. So the name is active transport. So we are discussing different mechanisms of transport across the cell membrane. Cell membrane is not like from which everything can pass. It has got different, stopping mechanisms or barriers based upon which it stops anything which tries to pass through this. This famous structure of the cell membrane, which is called lipid bilayer. It is cell membrane, it is made up of phospholipids. And there is a hydrophilic end and there is a hydrophobic end. Water molecules can pass, but the water-soluble substances cannot pass. Lipid-soluble substances can pass easily. So anything which can be dissolved by the lipid, it can pass through the cell membrane easily. But the water-soluble substances, they cannot pass. This is a barrier. This is called a barrier. So nothing can pass easily through the cell membrane because, it is a lipid bilayer which is hydrophobic. It does not like water-soluble substances. it is tightly packed. It is not like this, but it is like this. Yani, tightly packed. It stops anything to pass through it. And finally, there is a charge on it. Inside of the cell membrane is negative. Outside of the cell membrane is positive. So these three factors. So negative charge is repelled. Positive charge is pulled. I repeat, cell membrane is a barrier. It does not allow everything to pass through it. I said there is one rule. Lipid soluble substances simply pass through the cell membrane, but there is one condition. That is, the concentration gradient must be maintained. If for example, oxygen or carbon dioxide. If carbon dioxide is more inside the cell and it is less outside the cell, it can easily pass through the cell membrane from more to less. If oxygen is more on the outside of the cell and it is less inside the cell, then oxygen can easily pass through cell membrane anytime from the high concentration to the low concentration. So this is the concept of the transport across cell membrane. Now it has got some protein channels. There are some proteins in the cell membrane which allow some substances to pass. But again, this protein, it has got gate. Like we have got the door here, but this door is closed. But we can open this door. If this is a classroom for second year PT students, second year RC students cannot enter here. Emergency medicine students cannot enter here because outside is a guard sitting who just recognizes, or you punch your card of second year PT and you can enter the door. Just like that, we will discuss these small points as we go further, inshallah. So I repeat on this slide and then I will give you a writing. The lipid soluble substances pass freely. This is simple diffusion or passive diffusion. There are some channels through the proteins through which substances can pass along the concentration gradient. Some substances need vasta. They need someone to help them. For example, glucose will need insulin to open the gate and make it go along the concentration gradient to the side where there is less glucose. But some substances, they need active transport. They break ATP, convert it into ADP. This liberates phosphate or energy. And this active process throws out sodium from inside outside, because outside sodium is more, inside sodium is less. Or it brings back potassium from outside inside against the concentration gradient, because potassium is more on the inner side. So whenever we talk of active transport, we say active transport is against concentration gradient and it uses energy. Passive diffusion, it is along the concentration gradient, it does not need energy. It does not need energy. And this we will put in detail in the next 10 slides. We will go slowly, but please do not miss the slides for your exam. Read the slides for the exam. Prepare the slides for the exam. All right. So, what are the two major types of transport across cell membrane? One is called passive transport and the other is called active transport. See, the second name for passive transport is diffusion. So, it is net movement of molecules and ions across a membrane from higher to lower concentration, or we say down the concentration gradient. Does it require energy? No, it does not require energy. The other is active transport. Now here it is special. Number one, it is movement against the concentration gradient. It goes to the region of higher concentration, exactly opposite to the diffusion. Number two, it requires ATP or energy. Number three, it requires carrier protein. So 3 points to note here, 2 points to note here in the diffusion. It is, along or down the concentration gradient. This is against the concentration gradient. This does not require energy, this requires energy. This requires carrier protein also, yani active transport. Now, same description I have given over here. What is the rule in the passive transport? The rule is this: it goes from the higher concentration to the lower concentration. What if the concentration is equal on both the sides? There will be no diffusion. So this is called tawazun, or this is called equilibrium in the scientific language. So my dears, diffusion has some properties which I discussed with you. It is not needing ATP, it does not need carrier protein, and when the equilibrium is reached, then the diffusion it stops. When both the sides they have got equal amount of the substance. There is a special type of diffusion which does not need ATP, but it needs the carrier protein. This type is called facilitated diffusion. This is different. See, this word is now changed. Before it was passive transport or just simple diffusion or passive diffusion. Now this is facilitated. Saleh, can you translate the word facilitated? Where someone helps. Where someone facilitates or helps. Who helps in facilitated diffusion? The carrier protein it helps in the facilitated diffusion. Yani, some special molecules have got some special proteins which recognize them and they allow these substances to pass, but there is no use of energy. So the protein carrier it helps. This is called facilitates. And it does not require energy, but still it is from higher concentration to the low concentration. That is why it is called diffusion. This carrier protein is same like this in the active transport. But what is the difference? The difference is that active transport needs energy. It needs ATP. Facilitated diffusion is from higher concentration to lower concentration. Active transport is from lower to higher concentration. Facilitated diffusion need carrier protein. Active transport also need carrier protein. But facilitated diffusion does not need ATP. But the active transport it needs ATP. Why it needs ATP? Because it is from the lower concentration to the higher concentration. So we come to the active transport and give an example. Yani just not wasting the time. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 1, 2, 3, 4, 5, 6, 7, 8. This. You see, this is trying to go to the higher concentration. What is the name? Active transport. If it is coming from here to this side, it will be diffusion. So now this is going in the reverse direction. Why is it required? It is required sometimes because like we generate action potential. So for generation of action potential, sodium first goes into the cell and causes depolarization. So this sodium must go out. But how will it go out? Because outside is a lot of sodium. So it is against the concentration gradient. So it will use the active transport. So what is active transport? Active transport is when the cells move material against the concentration gradient. It requires energy and it requires special proteins which are called pumps. It requires special proteins which are called pumps. Now we have got some special transport mechanisms which I will name and you will prepare them for the quiz or the exam. One of them is called endocytosis. What is endocytosis? Endocytosis is this, that something comes near the cell and the cell membrane, it goes down and it brings it inside it, and then these two parts they unite and they take the thing into the cell. So this is eating, cell tries to eat something. Now this thing can be small and this thing can be big. I'll go to the next slide and I'll explain. Endocytosis is the process by which cell absorb molecules, such as proteins, by engulfing them. It is used by all cells of the body because most substances important to them are large polar molecules that cannot pass through the hydrophobic plasma or cell membrane simply this substance, big substance, which cannot pass through the cell membrane, it touches the cell membrane goes down, it makes a vesicle around it, takes it into the cell, and then this is eaten up. It is two types. One where very small substances, they are there, and these small substances, they are taken up. So it is a form of endocytosis in which small particles are brought into the cell. And the other one is eating. We say this is drinking, this is eating. So big substances like bacteria. For example, your white blood cell, neutrophil, it takes up full bacteria, many bacteria, and eats them and destroys them. So phagocytosis is the type of endocytosis in which big solid particles are engulfed and transferred into the cytoplasm by the cell membrane. My dears, we have short answer questions as well. So if we ask you a question, what are the two types of endocytosis? So you can write the two types of endocytosis are phagocytosis and pinocytosis. Alright? So this is it. Now, exactly opposite to it. See, this is endocytosis, which is taking in a substance. What will be the name of throwing out some substance? It is exactly opposite to it. It is called exocytosis. So exocytosis is throwing out a substance, for example, secretion. This cell has produced a hormone. So this is coming in a vesicle near the cell membrane. It joins with the cell membrane, and this thing goes out. So this is called exocytosis, the process by which cell moves the contents of secretory vesicle from inside the cytoplasm to the outside of the cell membrane. So this is the exactly opposite to the endocytosis. Now, why we are discussing this topic? Because of this problem that everything cannot pass through the cell membrane, the inside of the cell is different from outside of the cell. So this leads to a difference of constituents between inside and outside of the cell. So intracellular fluid is different from the extracellular fluid. Now, what are the main differences? We will discuss this slowly. Number one, our concept must be clear that if we have got about 40 liter or 60% of our body weight as water, out of this, about 25 liter is present inside the cell. This is your MCQ. Where is our maximum body water? Inside the cell or intracellular fluid. And about 12 liter or less than half is, in the space which is outside the cell. 3 liter is in the blood vessels. So this 12 plus 3, this makes extracellular fluid. So extracellular fluid is 20% of body weight, intracellular fluid is about 40% of body weight, 20 × 40 makes 60. So how much your body weight is water? It is about 60%. So 60% of your body weight, this is water. So where is body water more? In the intracellular fluid. This was point number one. Now, sodium, just a few examples and then a slide. Sodium is more in the extracellular fluid. It is less in the intracellular fluid. Potassium is more in the intracellular fluid, and potassium is less in the extracellular. Wherever sodium is more, chloride is more. So chloride is more in the extracellular fluid. Proteins are produced in the cells. Proteins are produced in the cells. So proteins are more in the intracellular fluid. Phosphate is more in the intracellular fluid. Bicarbonate is more in the extracellular fluid. So we will remember just a few things, not many. Not many, please don't worry. One, I want you to remember that sodium is more in the extracellular fluid, it is less in the intracellular fluid. Two, I want you to remember that potassium is more in the intracellular fluid and less in the extracellular fluid. Three, I want you to remember that proteins are more in the intracellular fluid. So proteins, high concentration of proteins in the intracellular fluid. Alright? Where do we get our food from? We get our food from extracellular fluid. What do you mean? I go back. What is this? What is this? This is blood. This is plasma. We take food, it goes into our intestine, stomach and intestine. From where it is absorbed? From the intestine. Where does it go? It goes into the blood. What is blood? Blood is extracellular fluid. Sahi? From here it goes into the interstitial fluid. So glucose, protein, whatever we eat, lipids, and then if the cell needs, it is transferred to the cells. So we make our concept clear that the food is more in the extracellular fluid, like oxygen, glucose, fatty acids, amino acids. Okay, so just this is the difference between. Now, what is the advantage of this difference? See, I start once again from only 2 points. See, the sodium is more outside, it is less inside. Potassium is more inside, it is less outside. So what is the advantage? The advantage is this, my dears, that this makes our cell membrane potential. See, if everything is equal, yani 10 plus and 10 minus outside, 10 plus 10 minus inside, this is a dead cell where everything is equal, and there is no current. See, this membrane has no potential, as concentration of charges is equal on both sides of the membrane. Now see the difference. Please keep this in your mind. 10 plus 10 minus equal 10 plus 10 minus. No potential. This is a dead cell. Now see the difference. This has got 15 plus 10 minus. This has got 5 plus 10 minus. Are the two sides different? Yes. So this membrane has potential. What is separating it? The membrane, the cell membrane. So Allah subhanahu wa ta'ala has made a system. See, the same 15 and 10, yani 5 and 10, they are not equal. But plus pulls negative. Plus pulls negative. So plus it comes on the outside and negative it comes on the inside. Try to understand this thing. So sodium is more on the outside. It pulls negative charges, but they cannot cross the cell membrane. So we say that the charges are separated by the cell membrane. In the rest of the places, everything is balanced. Plus, minus, plus, minus, plus, minus, plus, minus, plus, minus. Plus, minus, plus, minus, plus, minus, plus, minus. Can you see? Everything is balanced. But these are 5 extra. You see? Some extra here. 10 negative make pair with 10 positive. So 5 positive extra. 5 positive make pair with Zawaj. They make pair with 5 negative. So 5 negative extra. So these 5, 1, 2, 3, 4, 5 and 1, 2, 3, 4, 5. They cannot meet because they are an extra intracellular fluid separated by the cell membrane. So my dears, we say the membrane has potential due to difference of charges on the two sides of the membrane. Yani, if I go back, please don't be confused. Very simple thing. If the charges are equal on both the sides of the membrane has no potential. It cannot do anything. But if the charges are different on the two sides, the membrane has potential. The charges are not equal on the two sides, intracellular fluid and extracellular fluid. So this membrane has potential. Now this membrane which has potential and it is resting, we say it is resting membrane potential. This membrane is negative inside. So please remember, inside of the cell membrane is
Negative.
negative. What is resting membrane potential? It is a potential on the cell membrane when the cell membrane is resting, when our muscle is resting, when our neuron is resting. Then it has the charge which can be changed. So, what are the causes for membrane potential? We have discussed these factors and I'll put them here one by one. Number one, the cell membrane has slightly more positive ions on the outside and slightly more negative side on the inside. More negative on the inside. This difference produces a voltage which is called membrane potential. Now this is for your quiz. This was for understanding and now this is for quiz. This membrane potential is called resting membrane potential, and the membrane potential is measured in millivolts. And for example, the resting membrane potential in neurons, this is minus 70 millivolts. Minus 70 millivolts. Now, two factors are more important in production of membrane potential. The first is the unequal distribution of ions. That is why I told you, remember, sodium more on the outside, sodium less on the inside. Potassium more on the inside, potassium less on the outside. So unequal distribution of the ions across the membrane due to their selective movement through the plasma membrane. And next slide I will put another point here. Number two is the sodium-potassium pump, which is active transport. What does it do? We will see. So what is point number one? Now we'll explain point number one, an unequal distribution. How this unequal distribution takes place. So electrical charges are present on the ions. The ions primarily responsible for the generation of resting membrane potential are sodium, potassium, and the negatively charged proteins, which I told you they are more inside. Sodium ions are greater concentration in extracellular fluid. Potassium ions are in much higher concentration in the intracellular fluid. Proteins or anions are found only inside the cell. These excess anions trapped inside the cell help the inside to become more negative. All these points, they are MCQ or they are EMQ, extended matching question. This slide is very, very important because I made summary of 10 slides on this slide. And then a very important point, sodium and potassium can passively cross the membrane through protein channels which are specific for them. The membrane is 50 times more permeable to potassium than to sodium. This point is so important that I have made it bold and underlined. That is, we will ask you how much is the permeability of potassium as compared to sodium. What is the answer? 50 times. 50 times. Okay. So there is a continuous tendency of sodium to diffuse in and potassium to diffuse out because of the concentration gradient of each cell. So this was about point number one. Now the point number two. What is sodium-potassium pump? So sodium-potassium pump is this, that there is a carrier protein. It is a type of active transport. There is an enzyme ATPase to break ATP. So what is this? It throws out 3 sodium from inside the cell out. And it takes back 2 potassium. What is the charge on sodium? Plus.
Yes.
How many plus going out?
3.
3. What is the charge on potassium?
Plus.
Plus. How many potassium coming in?
So
2. So 3 go out, 2 come in. Inside will become negative. Because more positive is going out. 3 going out, 2 coming back. So what is sodium-potassium pump? Sodium-potassium pump is a type of active transport it pumps 3 sodium out and brings back 2 potassium. Same. This results in the presence of more positive charges in the extracellular fluid, and this generates part of the membrane potential. Okay, so there are some relevant videos over here which you can see to learn and understand these things. Can you answer my short questions? Number 1, name the 2 basic forms of endocytosis. Okay, yes, Mahmoud, can you tell? So, one is pinocytosis and the other is phagocytosis. So like if you get this in the short answer for the exam, don't be afraid. So this is a short answer question. It is not MCQ. So just write here 1 pinocytosis, 2 phagocytosis. Right? Okay, one example of the MCQ. The major volume of water in our body is where?
Intracellular.
Intracellular fluid. So you will make this, if you have got A and B and C and D on your sheet, you will make this B. Sahi? So this is an example. So we take a short break. Pause. Alright, Bismillahirrahmanirrahim. So our second lecture for today is the changes in the membrane potential. In the previous lecture we discussed the resting membrane potential and the reason for it. We discussed that the reason for it is unequal distribution of ions on the 2 sides of cell membrane. And number 2, sodium-potassium pump, which throws out 3 sodium, that is 3 plus, and takes back 2 potassium, yani 2 positive. So this means 3 minus 2. Inside becomes negative. So this is the story which we discussed. Now here we will revise your first semester thing of depolarization, repolarization, hyperpolarization, action potential, and a couple of new words. Like we'll have the word graded potential, and we'll have excitatory potential and inhibitory potential and action potential, of course. So we'll just pass through this small part of the membrane potential. So there are 2 types of electrical signals. One is graded potential, which can go for very short area, yani they are just local, and the action potential which are strong, and they can generate big impulse. So this can travel for long distance, like my brain orders my finger to move. So this is a signal which is coming from here to here in the muscle. So this means it is traveling long distance. Will it be graded potential or action potential? Action potential, because it is action potential which travels for long distance. Something touch on my here, just on my skin, very small thing, and this is not recorded by my brain. And Saleh tells me, doctor, there is small insect, bauda, mosquito sitting on your skin, and I don't feel it. Yani, it was not able to generate action potential. It just generates a small change in the potential. This is called graded potential, which goes for short distance or it dies locally. So these are the 2 types of changes in the membrane potential. Now, if the potential change, number 1, I told you that the resting membrane is minus. If you go to the positive side, this is called depolarization. If you go from positive to negative side, this is called repolarization. If you go below the resting membrane potential. This is called hyperpolarization. Yani the membrane become more negative. Let's make the concept again. If membrane becomes a little positive, yani minus 70 is more negative, minus 50 is less negative. So it becomes positive. This is depolarization. If it comes back to negative, this is called repolarization. This is resting membrane potential. This is resting membrane potential. If it goes below the resting membrane potential, it is called hyperpolarization. Alright? So, a stimulus. What is a stimulus? A stimulus is a change in the environment, internal or external. For example, inside my body, blood glucose increases. This is stimulus. For example, inside my body, calcium decreases. This is stimulus. That is why we discussed homeostasis. Or, for example, outside, the temperature increases. This is a stimulus. Or I think that I have left my car key in my car or somewhere else and I become anxious. This is a stimulus inside my body. So a stimulus can be small stimulus which makes graded potential, or it can reach the threshold, yani the level where it generates the action potential. Saleh, can you translate threshold into Arabic? It is the level where something happens. Yani, for example, you are trying to make me angry and I am not becoming angry. First, you make some movement. I don't become angry because it has not reached the threshold, my anger level. Then you put your leg in front of me and I suddenly become angry. You see? So because you have reached the threshold, had, it is the level. So if it reaches a level, then it can generate action potential. So the changes in resting membrane potential, number one is depolarization. The membrane potential is reduced from RMP, yani it is decreased or it moves towards positive. I am explaining this slide because many students still they are not able to prepare MCQ from this. So these four topics here, resting membrane, depolarization, repolarization, hyperpolarization, these four. 1, 2, 3, 4. This I have put on the next slide. What is resting membrane potential? I have discussed it with you in detail. It is the change of the cell membrane when it is not stimulated. Sorry, it is the charge of the cell membrane when it is not stimulated. It is at rest. It is negative inside as compared with the outside. What is depolarization? The membrane potential is reduced. It moves towards positivity. It moves towards zero. What is repolarization? It is the return of the membrane potential to resting membrane potential after being depolarized. It is returned to the resting negativity. So I go back. This is repolarization. Yani from negative 50, it has become negative 70. Yani it has become more negative. And then what is hyperpolarization? Hyperpolarization, the membrane potential is greater than the resting membrane potential. That is, it is more negative than the resting membrane potential. So it is, you see, this is resting membrane. It is more negative. It is going to minus 80. Resting membrane is minus 70. So this is called hyperpolarization. So this I have put for MCQ. Okay? So this is very simple. I will give depolarization is which of the following? And I will put 1, 2, 3, 4 definitions. Or I will put what is hyperpolarization? And I will put 1, 2, 3, 4 and you have to choose. So prepare right here important slide for MCQ. Alright. So as I told you on this slide that there is graded potential and there is action potential. So now we are going to organize this. So what is graded potential? Graded potential is a localized change in the membrane potential. It's very small. so small that it does not move further. What is this diagram telling us? Resting membrane potential. Unbalanced charges distributed across the plasma membrane that are responsible for the resting membrane potential. Inside is negative. Outside is
Positive.
positive. This is a sodium channel which is
Closed.
closed. Now see, when the stimulus is applied, this causes entry of sodium. What is the charge on sodium? Plus.
Plus.
When sodium goes in, what will happen to this negative? It will become positive. It has become positive. But because the stimulus is very small, so this is not going to this, this is not going to this. So we say active area depolarized. This is a graded potential, but the area around the stimulus, it is inactive. Inactive resting potential. Inactive resting potential. So what do we call it? A localized change in the membrane potential. I go back. So a localized change in the membrane potential is called graded potential. Okay, so this is title and this is next. Now, it is possible that this positive makes this area a little positive. You see, this is changing the color. Can you note the change in the color? Yani, this is making previously inactive area depolarized. Previously inactive area depolarized. See, I try to make animation. I go back. Only this area is stimulated. Now, this is spread to the adjacent area. This is spread of depolarization. This is trying to stimulate the area, but the next area is still inactive. Now, how powerful is the stimulus? This is the story. If the stimulus is very powerful, then it will spread very fast and it will become action potential. If the stimulus is very small, this will remain in a very small area. It will not be spreading. It will, the mosquito sitting here, I could not feel it. Someone tells me, doctor, there is a small mosquito sitting here. Remove it. Graded potential. So graded potential spread by passive current flow. It is not action potential. It is directly dependent upon the strength of the stimulus. If the stimulus is small, the potential is small. If the stimulus is big, the potential is big. If the stimulus is long, the potential is long. If the stimulus is big and long, the potential is big and long. You understand?
I understand.
One is this, I touch you. You see, I touch some person like this, small stimulus. The other is, I touch and press and keep pressing. So long stimulus, powerful stimulus. Third is, I punch.
Yes.
You understand? Powerful stimulus, not long,
Yes.
but powerful stimulus. Four, which is very bad. I take a knife and I cut the skin.
Powerful lung.
You know, it is powerful and long.
Yes.
So this is the difference between the strength of stimulus and the graded potential. Can you name some examples of graded potential? Yes. Post-synaptic potential, like excitatory post-synaptic potential, inhibitory post-synaptic potential, receptor potential, end plate potential, pacemaker potential, slow wave potential. Why did we put this slide here? Because in physiotherapy, they will teach you a special thing. There is a purpose that we are doing this special course. They will teach you how to stimulate a dead limb. A person has got shell or paralysis. The leg is not working, but the nerves are there. So you will be giving some stimulus. Now this stimulus is not coming from the brain because there is damage in the spinal cord or somewhere. But you will try to stimulate the nerve with electric current. How much current to give? How long current to give? So these names you will use there. Receptor potential, end plate potential, pacemaker potential, and many other type of potential. And I will not go into detail of these things. These are the examples of
graded.
graded. Is there any action potential in them? No. Can they travel in the nerve? No. How are they different from action potential? Now, action potential, we have seen this diagram before as well. It is having a very strong stimulus which reaches the threshold potential. When it reaches the threshold potential, it is no more graded potential. It becomes red action potential. Suddenly sodium channels open, a lot of sodium comes in, it becomes maximally positive. Then the sodium channel close, potassium channel open, it becomes repolarized. Then it becomes resting membrane potential once again. Let's explain these steps which I told you just now. So we have to discuss number one, the triggering event which opens the voltage gated channel. I go back. This is the triggering event. The stimulus. You know, I told you this is a small stimulus, and this punching is a big stimulus, and this cutting is a big and long stimulus. Hurt, harm. So triggering event. So remember this diagram in your mind, okay? So what is action potential? They are brief, rapid, large changes in the membrane potential during which the potential actually reverses so that the inside becomes positive. Negative 70, 70, 70, 70, 30, plus 30. It becomes positive. Then it is brought back to normal. You see? And we will describe it on the next slide. So triggering the event opens voltage gated channels, depolarization, decreased membrane potential. This leads to opening of some voltage gated sodium channel, influx of sodium, and this is called depolarization. This is positive feedback. Triggering event, opens sodium channel, opens more sodium channel, makes it positive. This is called depolarization. Sorry? This is example of action potential. What are the parts of the action potential and what causes it? So I told you, please prepare the figures. Figures are important. Figures are important for the exam. So number one, rising phase. The name is depolarization. What is the cause? It is caused by sodium influx. That is, sodium is going into the cell. What is the falling phase? Falling phase is called repolarization. What is the cause? The cause is by potassium efflux. Efflux means potassium going out. Influx means coming in. Sorry? So now we will describe it a little bit. Threshold potential, depolarization, repolarization, and as always, description. So that you prepare it for your MCQ. Full thing which I said up till now. I start from here so that you trust me. This is written there. This is written there. These are written there, okay? So I combined all this for those students who are lazy and do not prepare the figures. So same thing. So trust me, I will not read everything. A rapid change reversal in the membrane potential in response to a stimulus is called action potential. The changes in potential during an action potential occur as a result of ion movement. What is depolarization? Adequate stimulus, depolarization of membrane by sodium influx. When membrane potential reaches a certain level, threshold which is minus 55, rapid opening of sodium channel. From where I brought it? I brought it from here. See, exactly this thing. If depolarization occurs from resting potential of minus 70 millivolt to minus 55 or minus 50 millivolt, threshold potential will be achieved and action potential will be generated. So this same is on this slide. So now you trust me, okay? So likewise, what is repolarization? So at the peak plus 30, sodium channel closed and the membrane permeability to sodium decrease, potassium channels open and permeability to potassium increases, potassium moves out of the cell and this restores gradually the membrane potential. This is a YouTube movie for action potential. We have two types of excitable tissue in the body, nerve and muscle. What is a stimulus? I discussed with you. Stimulus is a detectable change in the internal or external environment of the body. Anything that has a potential to cause excitability. For example, it can be change in temperature. For example, it can be a sound. For example, it can be light which we can see. For example, it can be change in my blood glucose. For example, it can be change in my blood pressure. So this is internal and external, different examples. So it can be internal or external change. This change can be threshold stimulus or sub-threshold stimulus. You punched another person, threshold stimulus. Some small mosquito sit on some person's skin, he does not notice it, sub-threshold. Yani tahat the had. It is under the head, the threshold. What is excitability? What is the word excitability? It is the ability to sense changes within and around us and response. What is conductivity? Conduction. What is conduction? It is ability to carry a stimulus or a response from one part of a cell to another part of a cell. Like the nerve conducts. It brings action potential from my brain to my finger, for example. So this is conduction. And now what is left? See, what is the topic? Excitable tissues. So nerve conducts. What does muscle do? Contract. So the third thing is contractility. Contractility is the ability of cells to become short. You see, I make my muscle big. Actually, it is becoming short. I pick up something. It is becoming short. A wrestler or a weightlifter picks up weight. Actually, the muscles are becoming short. So this is called contraction. So we will slowly, inshallah, revise these things. What is the structural and functional unit of the nervous system? It is neuron. Okay, so neuron is the structural and functional unit of the nervous system. So, because the topic is action potential, so we finished on the action potential conduction. So there are two methods of propagation of action potential in the nerve. One is called saltatory or jumping, like a rabbit, arnab.
Yes.
It is present in the myelinated nerves. And the other is conduction by local current flow. So these are the action potential transport. What is saltatory conduction? Saltatory conduction is in the myelinated nerve fiber. Action potential jumps from one node to the other node, from one node to the other node. This is called saltatory. Saltatory means jumping. So it is fast. 2 points. Don't worry, it will be on the next slide as well. Number one, it is in the myelinated nerve. Number two, it is fast. Okay, so this is conduction of the action potential. So this display, it is telling the two types of the conduction. This one is found in the myelinated nerve. Current jumps, 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 jumps. The speed will be fast. Here, the current flows. There is no insulation, no myelination. It is continuously flowing. There is fluid outside. Some current is lost on the outside. So number one, it is unmyelinated. Number two, it is slow. Number three, if it is very powerful here, by the time it reaches here, it becomes less powerful because the current is lost in the fluid around the neuron. Here, less current is lost. Here, it is fast. What is the name? saltatory conduction. Alright. So I said, don't worry, it is written on the slide. So for those of you who do not want to remember from the diagrams, here is the slide. What is saltatory conduction? It occurs in myelinated fibers. Myelin is a phospholipid, does not conduct electric current, yani it is ghair muwassal, insulator. Myelin sheath is interrupted at various intervals, that is node of Renberg. Nerve impulses are conducted from node to node as ions can move only in these areas. So charges will jump from node to next node. This is known as saltatory conduction. This type of conduction is important because it increases the speed of conduction of nerve impulses and it saves energy since the activity is localized at the nodes. So this was about saltatory conduction. This is about the other one. That is the conduction by local current flow. That is now I am putting description for this one. This is saltatory, okay? Yes. So here, the stimulus is here. This area becomes active. This area is inactive. So this area stimulates the next. This area stimulates the next. So this area has stimulated the next. Yani, inside became positive. Inside became negative, but the next became positive. See? So this is the conduction by local flow. How is it? Where is it? It is in unmyelinated nerve fibers. Initiation of action potential in an area makes it an active area inside the membrane, becomes positive, and the remaining of the fiber is inactive with the inside negative. The generated action potential is conducted along the membrane of the fiber as a wave of negativity. So this was about conduction, and 3 slides more for today. One type of nerve fibers. This is to prepare for an MCQ, and I have made it on 3 slides. So there are different types of nerves. Just remember one rule. If a nerve has got big diameter, if it is thick, the speed will be fast. If the diameter is this much, the speed will be 80 to 120 meters per second. If it becomes small, the speed will become 5.5 to 2 meters per second. So simple rule, if a nerve is unmyelinated, the speed of conduction will be slow. If a nerve is thin, the speed will be slow. If a nerve is myelinated, speed will be high. If a nerve is myelinated and big diameter, the speed will be very high. Okay, so this is the basic concept. So we have put it on a diagram. These 4. See these 4 names: alpha, beta, gamma, delta, C. It is put here. So alpha, beta, delta, C. Diameter, 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 decreasing. See? Speed in yani more, less, least. In C fiber it is less. These take up pain. These take up proprioception or the position. These are myelinated, and C is unmyelinated. Now, many MCQs will be made from this. Very important slide. Okay. Which one of the following is unmyelinated?
C.
C. C, but the rest 3 are myelinated. Which one of the following, yani alpha, beta, delta, and C type of nerve fiber has got the maximum speed?
Alpha.
See, alpha. You understand?
Yes.
Which one of the following is for proprioception?
Alpha.
Alpha. What is proprioception? The position. See? Yes. What is the position of different joints of the body? So this slide, yani it is to remember. Alpha is the plane. Beta is the racing car. You understand? Which is very fast, but its speed is less than the plane. You understand? And delta is the bicycle. It is very fast, but it is not as fast as the train and the racing car and the plane. And C is walking. You understand?
Yes.
Which is not as fast, and we stop here. 69.